Hello everyone, thank you for attending. And my name is Paula Llanos. I'm part of the Simini Lab. I'm a postdoc there. And today in this workshop, we will work in a 3D segmentation pipeline. And our TAs will be Esteban and Sugania and Chatavisha. They are also part of the Simini Lab. So first, I will start with an overview of CellProfiler, which if you already attended yesterday for to introduction of CellProfiler, I think you will already know, you already have some knowledge about that, so but I will go fast for it. And then we will continue with a hands-on exercise on 3D segmentation. As I mentioned before, if you haven't downloaded yet CellProfiler, you can download from this link. And also to download the material, and you can download the material here in this link. And I think it will be available in the chat, right? So you can see there. So how CellProfiler build is building, how CellProfiler works is building pipelines. So the pipeline is just a it's a pipeline, right? Which is built with modules. So, and this is how this looks like. So for example, if we have a pipeline, then we will start the pipeline loading our image and set some setting, right, to load our image and then continue do some pre-processing on our image to do our aim, which is identify objects and to get some measurements, right? And finally, once we get our measurements, we will export the data. That is the main idea of how it's working CellProfiler. So the pipeline will build using modules. So each module will works with an input and an output. So it is something that you can name. So each module will receive an image and then will produce an image as well. And depend what is the aim of the module. So you need to name accordingly also. And when you enter to CellProfiler, you will see this main window. So this main window will have a left panel, which is the pipeline panel. This left panel will contain these 4 input module, which is just to set the data or the kind of data that you have, the image, and etc. And then this panel here will contain all the modules that we are using to build our pipeline. In the right panel here, you will see all the settings for each module. So for example, in this case, this image module will contain the window where you can drag your image to start the pipeline, right? To start the analysis. Something super important in CellProfiler is that we have here some question mark. In each setting that you will find a question mark. And that is super helpful to understand how is working each setting, how you can modify, right? So in the exercise here, and the exercise that we are working in this workshop, we will work in 3D for segmenting 3D image. So for that, we will have a set of 3 channels: one DNA channel, membrane channel, and mito channel, and how this looks like. So those are 3D, and you will see after in CellProfiler that you can go a slide between the slides in the between the each theta plane in the image, so you can see the whole 3D set. But we will contain in the exercise this image in the material. Okay. So, and the pipeline that you will build will look like that in the end. Is well, of course, that is always the module that you will find by default, the input module. Then we will include some modules to do the nuclei segmentation, also other module to do the cell segmentation, other to make some measurements, other to add some quality check modules to see if our segmentation is working well, and also some modules to export the data. Of course, we will in the hands-on exercise, we will go step by step in the setting and in each module that you need to add in the pipeline. So first, as I mentioned before, the pipeline always will start with the load image file, the load image file modules, and those are fixed, so you can extract all the information that is useful. That can be metadata that you want to include as a CVS, or also metadata that is contained in the name itself. You can also label your your image to group it and other option as well. And the input module, you will, as I mentioned before, you will have this panel and where you can double click and open the folder with your image, or just drag the image there. So when you do a double click in one image, you will have this display window, and where you can be able to, for example, with this tool, or with this specifically this one, you can, for example, measure approximately how is the diameters of the nuclei or the intensity of those of the nuclei. Then you can set the thresholding setting or watershed up properly. You will see, for example, here the diameter or the size of each object. So intensity, you can adjust the contrast, for example, if the signal is super weak. Also, you can adjust the contrast there to be able to see better. And when you start the pipeline, when you start CellProfiler, and you will find this panel empty, right? Because we didn't still, or you need to still add the module. So to add each module, you will find this button here, the plus button, and then you will see this window where the modules are categorized, but also you can also type the name of the module, and then if you already know what is the name of the module, you can just search typing there. And there is two buttons also in the main panel, which are very important to differentiate, because what we are using mostly is this button, Start Test Mode. That button will allow you to try the modules that you already add, or the pipeline that you build in one image. So when you are testing a pipeline, when you are building the pipeline and still trying different settings, you will use mostly this, because you don't want to analyze or try the pipeline in all your images, especially when you have hundreds or thousands of images. You don't want to use that, right? Just to test the pipeline. So when you are already set your settings and for each module, and you are already happy with the results, and then you try in different image, then you can go to this button, which is the button to analyze all the image that you have. So difference also, while you are adding modules, these all are modules that you can add, and each module have these different buttons. So for example, this green one is to run this module specifically. This checkbox is you can use if you want to include that module in the pipeline, or you want to skip that module during the pipeline. Also, you will find this error in module. For example, this signal, you can put your mouse on that symbol, and then you will see why this module brings you an error. Also, there is a pause symbol here. This is to when you want to run the pipeline, and then you want to run the pipeline until this module and see how it's performing the setting until that module. And also this little eye, you can open or close the eye, depend if you want to see the output of the module or you don't. Also, this warning message, this, for example, this is usually you will find when you want to add this export to spreadsheet module, but that warning message is because in this case, this module just work when you analyze all the whole batch, not just in, so that you will find usually when you are in test mode model. And finally, these two buttons. So this button is to run all the modules. So when I am here, for example, and I just hit run, then all the modules will be running. Or if you want to run just one module by one module, you can just hit here instead. So, okay, when you already load your image, right, and you do some pre-processing, what we want to look for in the pipeline is to identify some object, right, to segment our objects to be able to measure and get some measurements and export the data, right? So, and it is something if you already attend to the introduction to CellProfiler yesterday, or if you are already use CellProfiler to segment some 2D image, you will see that to do that segmentation, CellProfiler have already some modules which are the name of those is identify primary object and identify secondary object and also tertiary object. Is in those modules we are using to do the segmentation very straightforward, right? So in general, identify primary object is used to, for example, if we have a membrane and nuclei signal, we will be able to segment the nuclei in this case, and then this mask of nuclei we can use to in the identify secondary object to use together with the cell signal to segment the cell, and the cell will be segmented in the places where are the nuclei. That was super straightforward in 2D. However, if you want to do something similar in 3D, those modules are not able to use for 3D image, then we need to do some modification in the pipeline. So it is what we are working today. So to work in 3D with 3D image in CellProfiler, first there is some, or of course in the tutorials there is more information also about how to input your 3D image. But in general, what is the more maybe easier way to use or to analyze your image is using that TIFF extension image and load each channel separately. And also, while we are adding modules, you can see in the help mark here, each modules you can check also if that module support or don't support 3D. So for example, identify primary object, which is the one that we usually use in 2D, doesn't support 2D, or if, for example, this median filter, you can see here that information. So it's something that also you can explore before to add a module. So for segmenting 3D objects, as I mentioned, we are not using this time identify primary objects or secondary or tertiary. We will use threshold modules to image segmentation, and then a watershed modules to identify each object separately. So just to have an idea about how will be our workflow for this exercise, we will start do some image processing in each channel to segment. In this case, I just put a nuclei object, and we will do some processing, then segment, then identify each object as I mentioned with the watershed, and finally get some measurement in our object. So in these thresholding modules, it's important, of course, know right what mean that. So when we are applying thresholding, is just differentiate what is the foreground and what is the background in the image. So to do that, you can set that threshold manually. Like for example, in the image we have this histogram. Then if you set the threshold here, for example, almost all the pixels will be part of the background. For example, these pixels are a very lower value of intensity, which mean are more in the background. So you can set the threshold depends where you want. So this, for example, intensity are allowed to the pixel that correspond to actually nuclei signals. So you can depend how you set or where you set your threshold, you will see different performance in the segmentation. But of course, you don't need to do manually. You can use just mathematical methods like which are included, and you will find that setting in the threshold method, like minimal cross entropy, also robust background. So, and also all those methods that you will find there, you will find also a question mark where with some more explanation about how those are working and which one you can use depending on the image that you have. And once we already differentiate what is the background and what is the foreground, we are, for example, here interested to actually identify objects separately, to declump object or just identify each object as an individual object, what we call instance segmentation, right? So to make that, we will have this intensity, this image like or 2D slice, right? And what we will do is apply a watershed, which get this image as a 3D representation of the intensity. And this is like just imagine like this 3D image in a swimming pool that you will fill water, and then the water will run through the valleys, and then those valleys will in the end correspond to the places where the object will be separate to the others. And those objects can be separated or declumped based on the intensity and also based on the shape. So also you can set, for example, in general for nuclei, because it has a very defined shape, you can maybe try that mode. And also once finally we identify already our objects, then we are ready to get some measurements, and there is a lot measurement that you can add. We will see in the hands-on part the modules measurement. You can measure intensity distribution, object size, colocalization. So different modules are available in CellProfilers that you can use. And once you have the already measurements done, and then you want to export, of course, is the final goal, right? To export all your image or measurements that you did. You have also a couple of ways to make it. So if you want to export the image, for example, the image that you generate for quality control, to have this there, or the mask or other kind of image, you have this save image module. And also if you want to export all the data that you measure, you can export the spreadsheet or export to the database in case that, for example, you want to analyze further in CellProfiler analysis, or if the amount of image that you generate is too big, then also you can export using SQL format. And finally, when we're done with the pipeline, we want to, if we want to share and we want to save for ourselves or share to others, there is two different options. You can save the project in CellProfiler, which contain also the path of the image, which is very useful if you want to work with the image in your computer. But for example, if you want to share that pipeline to other collaborators, then it's better, and I will recommend to export that pipeline as a CPpipe format, and then just that pipeline will contain just the modules that you add without the local path that you were using for your image. And that is the one way to segment in 3D, but also, and yesterday also it was another workshop about how to use those plugins that also will allow to segment in 3D, run CellPost, StarDisk, Elastic, that also are available in CellProfiler, and you can try those to segment your objects. And if you need more help, there is also more tutorial available of to segment in 3D, and also something that we encourage to do is if you have any questions in CellProfiler, we are very active answering in the forum, in emergency forum. So we really push the people to write the question there, and then we can answer any question that you have. And also twice a year we have opened some office hours if you want to discuss something with us, something of your pipeline with us. So you are very welcome to do that. And then now we are starting the hands-on part. So do you have any questions so far before to show you the tutorial? Okay. So we will continue. This is the folder where you will find the material. So this is a tutorial that you are following. What I recommend is download this 3D monolayer start pipeline. This is the pipeline that we are using to start. So it's so when you have you already open CellProfiler here, for example, and you have your download your pipeline start pipeline, you just drag that there, and then you when you load that pipeline there, you will see that those input module are already set. So I recommend start with that pipeline, then continue with the tutorial. In the tutorial also, you will find information about how input the image in and how set those modules there. But for time purposes, I already upload that pipeline with those modules already set, so you can skip that part. You can read and review and check how those modules were set, but you can just skip. So here in this importing data in CellProfiler, those modules, as I mentioned, are already done. So you can continue in this part, in this section of the tutorial, find objects. So you can continue there, and this tutorial have everything step by step, but still we will split now in different breakout rooms, and then with the TAs and me, we are going around the breakout rooms to see that everything is okay. And also we ask you that someone in each breakout rooms share the screen, to then everyone in that room can follow and comment, and then you can build the pipeline together. That is the main idea. So just to repeat that, you will start the tutorial from find objects step. Any questions so far before to split to breakout rooms? No? Okay. I will stop share.
I'm making the breakout, like creating the breakout rooms right now. So.
Okay, cool. Thank you. Yeah, all material, the presentation, everything is in the folder, just in case.